Okay, let's move into cell biology, and we'll begin with cell structure, looking at how cells are ordered and how they function. Okay, so let's look a little bit at cell theory. Anton von Leeuwenhoek, in the early 1600s, he was responsible for developing the first simple magnifying instrument. And today you call that magnifying instrument a, yes, correct, if you said a microscope. So he was responsible for the earliest um, type of microscope. Then Robert Hooke in the 1600s, also, he was the first person to actually coin the phrase cell when describing the structure of living things. He said that the cell um, of a monastery resembled what he was seeing in living things. For example, he got this idea because when the cork was sliced very thinly, the cork resembled the cell of a monastery. So it was more of a rectangular type structure. So he said that the cell um, of a monastery resembled the structure that he was actually visually seeing underneath the simple magnifying instrument. Then Slyden and Swan in the 1800s, they said all living things are composed or made of cells. So every single thing that we consider to be living is made up of cells. And then finally, Pasteur and Virchow in the 1800s, they said, well, there's no such thing as spontaneous generation of life. And how they came up with the idea that there's no spontaneous generation of life, again. So Schleiden and Swan, they took flask, and in the flask they put broth, and they knew that bacteria would thrive in this broth. Um, So you have colonies of bacteria growing in the broth, but they, if you remember Pasteur's name, he's famous for what? Pasteurization. So he boiled the broth. So these are my flames representing him boiling the broth. So once he boiled the broth, we knew that we had a bacteria free container. So they let the um, container sit out, and what they found was that the bacteria, once examined, had returned to the container. So now we got all these colonies of bacteria growing again. So the idea was, okay, does life just spontaneously appear out of nowhere? And they said, well, no. They believed that because of gravity, the bacteria was settling out of the air. So, in order to either prove or disprove their theory, they redesigned the flask. So they went through a flask redesign. So they created this crook neck flask. Okay, so again we've got this broth and it's got bacteria in it. Here's the opening. But anyway, they've boiled it as well. So now we've got this bacteria-free broth, and the question is, did the bacteria come back in the container? So the answer to your question is yes, but was it in the broth? The answer to that question is no. So I'm going to draw my flask over here since I've ran out of room. And what they found was that the broth remained free from bacteria. Okay. But because of gravity, the bacteria had settled out in the necks of the flask. So it settled out here, settled out here, okay, here. But before the air could reach the broth, all of the bacteria had settled out. So this is how they came up with there is this concept that there's no spontaneous generation of life. So in other words, life comes from life. All right. So let's keep moving. All right. So if you remember from the previous chapter, we talked about some of the major macromolecules and some of the things that we'll find in cells, and then also other biologically important molecules. Well, we knew one of them was water. Water is going to make up 70% of the cells that we're going to look at, but it says the other 29% of the cell are going to be those four major macromolecules. And if you remember what they are, they were carbohydrates, lipids, proteins, and nucleic acids. And then you have another 1% of minerals. Okay, so the two major types of cells, going back to chapter 1, we either had prokaryotic cells and we had eukaryotic cells. So if you remember that, we had prokaryotic cells, and if you go back to those three main domains, the prokaryotic cells included who? Bacteria and archaea, and then the eukaryotic cells was eukarya. Remember, we had 4 kingdoms listed under that, but let's go into the prokaryotic cells. All right, so for prokaryotic cells, we know that these guys have a what we call a nucleoid or a primitive nucleus. The reason why we said it was primitive, if you remember, the word prokaryotic means that it lacks a nuclear membrane. It lacks a nuclear membrane. These guys are small in size, especially when you compare it to eukaryotic cells. Generally, they have very few organelles. And in this category that we're talking about, prokaryotic cells, we include bacteria and then a specialized bacteria called blue-green bacteria. We also call them cyanobacteria. These guys are able to photosynthesize just like plant cells and release oxygen. Not all bacteria we know cause disease. Some bacteria are going to be what we call beneficial bacteria. And if you remember, if you've ever been to the doctor, you know you have a natural flora and fauna of bacteria, and some of those bacteria help and assist us in maintaining life. 3 common shapes that we want to talk about are and we'll talk about the common name as well as the scientific name. So the coccus, we want to refer to that as a sphere. So it's spherical, it's round. Then the spirillum, that's a spiral-shaped bacteria. We'll talk about this guy later, especially when we're talking about syphilis. We'll mention one of the spiral-shaped bacteria. And then the bacillus, that is a rod. So coccus is a sphere, spirillum, that's a spiral-shaped bacteria, and then bacillus is a rod-shaped bacteria. So let's move on to the eukaryotic cells. Eukaryotic cells, we know that they have a true nucleus. The word again, eukaryotic. So in other words, we know that this nuclear material, the DNA, is housed within a nuclear membrane. So it has a true nucleus. We know that these guys are bigger than prokaryotic cells. They have many organelles. So not only, we said prokaryotic cells have few organelles, so they have ribosomes, but for example, and then they have this primitive nucleus. But if we look at the eukaryotic cell, we know that they have mitochondria, and then they have some of the cells have chloroplast and Golgi apparatus and lysosomes, and the list just continues to go on, and we'll get into those a little bit later. And then the four kingdoms are Plantae, Animalia, Fungi, and Protistum. So in this category, we have four kingdoms that would include the plants, the animals, the fungi, and the protistans. All right, so let's move on to prokaryotic cell structure. Um, First, you'll have the cell envelope, and there are three parts that we want to look at with the cell envelope, and that is starting from the innermost, and that would be the plasma membrane, moving then to the cell wall, and then finally the covering, which is the capsule. Then if we looked at the cytoplasm, this is where this is the fluid portion of the cell. This is where we're going to find that nucleoid, ribosomes, and possibly some inclusion bodies. And then finally, then this guy also has appendages, the flagella, the fimbriae, and the sex pili. And we'll look at each one of those individually. So let's look at the cell envelope. And we're going to stop here because I can only record small amounts at a time. So, and I'm about to run out of time, so I will stop right here and then pick this up in the next video. Hope you guys have a wonderful day.